Hello, and welcome to this e-seminar, which will provide an introduction to the post-PCR analytical method of DNA melting curve analysis. It is assumed that you have a working knowledge of PCR and have viewed the previous e-seminar on PCR assay design. However, if you require additional information, please refer to the 2013 LGC publication, Good Practice Guide for the Application of Quantitative PCR by Nolan, Huggett and Sanchez, which is free to download at www.lgcgroup.com/NML. This e-seminar will provide you with a general introduction to PCR-based DNA melting analysis as a method for food authenticity testing, and provide guidance on how to design, implement and analyse PCR DNA melting assay data. Topics covered in this e-seminar will include the principles underpinning DNA melting analysis, DNA melting analysis workflow, designing PCR DNA melting assays, fluorescent dyes suitable for use with DNA melting analysis, generic values for thermal cycling parameter, examples of PCR instruments compatible with DNA melting analysis, examples of analytical software packages, and guidance on troubleshooting. In the following section, you will be introduced to the fundamental principles of DNA melting analysis. DNA melting curve analysis for PCR amplicons involves the amplification of a target DNA sequence in the presence of a fluorescent double-stranded DNA binding dye. The group of dyes employed for this type of analysis only fluoresce when bound to double-stranded DNA and excited by illumination at an appropriate wavelength. Post-PCR, the level of fluorescence in the reaction mix is monitored as the amplicons are gradually denatured, or melted, by progressively increasing the reaction mix temperature. During denaturation of the amplicon, any intercalated dye molecules are released and the level of fluorescence decreases. The maximum change in the rate of fluorescence decrease occurs at the melting temperature of the amplicons. This corresponds to the point in the DNA melt curve at which an amplicon remains 50% double-stranded. Differences in DNA sequence composition between amplicons generated for different species result in species-specific melting profiles. The application may therefore be used to help determine which species are present in a test sample. DNA melting curve analysis relies on the generation of melt curves. As illustrated in Figure 1, by plotting the normalised fluorescence against temperature, the point of inflection, which corresponds to the melting temperature Tm of an amplicon, can be identified. The level of resolution may be improved by plotting the negative first derivative for the denaturation curve as shown in Figure 2. The latter approach highlights the melting temperatures of any amplicons present in a reaction as distinct peaks. However, all double-stranded DNA molecules will generate peaks, including non-specific PCR products such as primer dimers. Primer artifacts may normally be distinguished by the fact that they generate low-intensity signals. Primer dimers occur as a consequence of DNA sequence self-complementarity between or within primer pairs. Secondary targets frequently result from the complete or partial duplication of genomic DNA sequences, or when an unoptimized PCR assay is employed. For DNA melting curve analysis, it is essential that the PCR assay specificity and performance are optimized. The DNA melting curve analysis workflow comprises 4 key stages. The first stage involves the design and optimization of PCR assays, which may include the identification of suitable pre-existing assays. PCR performance is optimized as for real-time PCR assays, but with the inclusion of the optimization of the concentration of a fluorescent double-stranded DNA intercalating dye. The second stage involves the PCR amplification of a DNA target sequence in the presence of a fluorescent intercalating dye. In stage 3, a DNA melting curve analysis of all the PCR products generated during thermal cycling is undertaken. The final stage involves the analysis of DNA melting curve data, which results in the calculation of the melting temperatures for all the PCR products present in the thermally cycled samples. In the following section, guidance and best practice for the design of DNA melting curve assays will be presented. When applied to food authenticity testing, the optimal targets for DNA melting curve analysis require a degree of DNA sequence difference between the species being tested for. Mitochondrial DNA targets have been widely employed for species identification, as many mitochondrial DNA sequences exhibit high rates of DNA sequence divergence. These include cytochrome B, cytochrome C oxidase subunit 1, and tRNA ATP synthase 8. Several open access DNA sequence databases are available, which can be consulted in order to obtain DNA sequence information for the target of choice and across multiple species. However, before undertaking the design of new primer sets for a DNA target, it is recommended that a search for and performance evaluation of existing primer sets should be performed. The Barcode of Life database, or BOLD, is an open access resource which contains DNA sequence information for a number of barcoding markers across a broad range of animal, plant, fungi and microbial species. BOLD is organised into a number of databases. The primary database comprises DNA barcoding sequence information for a number of verified specimens for each species entry. Complementary to this is a PCR barcode primer database. This contains DNA sequence information for all the primers that have been employed to generate barcoding sequences. The BOLD database represents a useful resource when searching for pre-existing primer sets for use in DNA melting analysis. Illustrated here is an example of a workflow for identifying existing PCR primer sets of DNA barcodes catalogued at BOLD. The first step involves searching for a species of interest in the main classification database. Each species record comprises multiple entries, each generated for a different sample. Each record details the DNA sequence used to classify the sample, as well as details of the barcode employed and the PCR primer utilised. Once a list of potential primers has been derived, they can be evaluated for use in a DNA melting curve approach, either in silico or empirically. Guidance on the design of primer sets for real-time PCR applications is provided in the e-seminar An Introduction to Designing Quantitative Real-Time PCR Assays, which is available to view using the link posted on the Food Authenticity Network website and via the link shown on the slide. However, for the purpose of designing new primer sets for DNA melting curve analysis, the following additional recommendations should also be considered. Firstly, prior to undertaking the design of new primer sets, the potential of the target DNA sequence to discriminate between amplicon sequence variants using DNA melting curve analysis should be evaluated in silico using appropriate software. Once a target DNA sequence has been selected, primer sets should be designed which can generate amplicons of at least 300 base pairs. This is because amplicons of at least 300 base pairs have a high probability of allowing the detection of DNA sequence variants, enabling high levels of PCR efficiency to be achieved. The primers should be between 18 to 24 bases in length, possess a G plus C content of 40 to 60%,
and have a predicted melting temperature of 55 to 65 degrees centigrade. A balanced distribution of G plus C and A plus T bases should be employed if possible, but sequences with 3 or more consecutive G or Cs should be avoided. The melting temperature for G plus C bases is higher than that for A plus T. The presence of consecutive G plus C bases can distort the uniformity of the melting process, particularly if they occur in the last 5 bases of the PCR product, and should be avoided. In addition, primer pair melting temperatures should be within 2 to 3 degrees centigrade of each other, exhibit no significant self or intracomplementarity, and possess no internal secondary structure. And finally, the primer binding sites should lack any appreciable secondary structure. Prior to designing new primer sets, DNA target sequences can be assessed for their suitability to discriminate between similar sequences with the use of appropriate software. A number of open access programs for deriving theoretical melting curves are now available, and include the Poland DNA and MeltSim packages. Poland is a web-based tool that can generate melting profiles for double and single-stranded DNA or RNA molecules or DNA-RNA hybrids. This allows the user to evaluate the discriminatory power of a DNA melting analysis in differentiating between known sequence variants for a DNA target. Alternatively, the open access application MeltSim can be employed, which performs a similar analysis to that of the Poland package. The web addresses for the Poland and MeltSim freeware packages at the time of recording are displayed on the slide.
DNA melting curve analysis relies on changes in amplicon melt kinetics, which occur as a consequence of differences in DNA sequence between closely related amplicons. The technique involves PCR amplification of a DNA target in the presence of a fluorescent, saturating, double-stranded DNA intercalating dye. The current generation of dyes employed with the technique are much brighter and less inhibitory to the PCR reactions than their predecessors, which included the DNA stains ethidium bromide and propidium iodide. The use of the new dyes at saturating concentrations enables superior discrimination of closely related amplicons. This is because the dye molecules that have been released during the melting process cannot relocate to free binding sites. Modern generation dyes now commonly employed include Evergreen and Cybergreen ER, and many reagent manufacturers now provide PCR master mixes which include a fluorescent, double-stranded DNA intercalating dye. A number of fluorescent, double-stranded DNA intercalating dyes are available, several of which are listed here. Dye manufacturers normally provide recommendations for employing their products for experimental purposes. However, the following guidelines provide some general recommendations. Firstly, optimal dye concentration should be determined empirically. As a rough guide, concentrations spanning an interval above and below the manufacturer's recommendations should be evaluated. Optimal concentrations will provide maximum fluorescence while not causing any significant PCR inhibition. These types of dye are photosensitive, so both stock and working solutions should be protected from exposure to light. Many of these dyes are also unstable once diluted, therefore it is recommended that fresh dilutions be prepared for each new experiment. In addition, because these dyes may interact with PCR reaction components, further PCR optimization may be required. Finally, as dye performance can vary with the use of different reaction reagents, it is good practice to evaluate the performance of more than one dye. Reaction setup for DNA melting curve analysis is similar to that for real-time PCR. However, as the presence of non-specific PCR products may have a significant impact on the final analysis, careful optimization of reaction components and conditions should be undertaken. The use of hot start Taq polymerase is recommended, as this may help reduce the likelihood of non-specific PCR products being generated. The table shown here illustrates the typical composition of a DNA melting PCR reaction and may be adopted for use with most primer sets. The volumes depicted may be used for guidance when performing an optimization. Starting with the volumes and concentrations recommended, the reaction can be optimized beginning with primer set selection, primer concentrations, dye concentration, magnesium concentration, and finally, the amount of template employed.
Thermal cycling profiles typically employed with DNA melting curve analysis are similar to those used with real-time quantitative PCR. Cycling may be performed using a standard 2-temperature protocol and for a duration of 40 cycles. An additional final denaturation step is performed following the amplification cycles, where the temperature is progressively raised from 75 to 95 degrees centigrade. During this step, the level of fluorescent signal is recorded. A significant factor in the generation of robust DNA melting curve data is the quality of the DNA template employed. Carryover of salt used during DNA isolation and purification can significantly impact on the specificity of the PCR reaction, as well as the melting temperature of the resulting amplicons. Many commercial DNA isolation kits employ buffers with high salt concentrations during the elution of DNA from specialized filters or columns. As well as salts, buffers which exhibit extremes of pH are also employed with many kits, particularly during the initial stages of DNA isolation. Both salt concentration and buffer pH can significantly influence the melting curves that are derived for a sample. Performing additional purification steps as a matter of routine is highly recommended. Precipitation of the template DNA using sodium acetate and ethanol, followed by the washing of the pellet with 70% ethanol and resuspension in nuclease-free water or low-salt buffer are generally recommended. Alternatively, template DNA can be desalted using a microcentrifugation device, such as a millipore YM30 microcentrifugation column. A large number of real-time PCR instruments are now available, which are capable of performing DNA melting curve analysis. This capability is frequently immediately available with some instrument makes, while others may require the purchase of additional functionality modules or software upgrades. The table presented here lists some of the instruments available with DNA melting analysis capability.
A number of software packages are currently available for analysing DNA melting curve data. Many of these have been compiled for use with specific platforms. However, a number of open access applications and macro-enabled spreadsheets are also available. Examples of available software are shown on the slide. Version 2 of the high-resolution melting software is currently distributed with some of the applied biosystems instruments available for purchase from Thermo Fisher Scientific. The analytical software Precision Melt Analysis is another example of commercially available software. It is distributed with and enables the analysis of DNA melting curve data generated on some of the Bio-Rad real-time PCR instruments. An example of an open access DNA melting curve analysis software package is also presented. The application analyse will perform many of the analysis undertaken by commercial software. It is free to download from the web addresses presented on the slide and which was correct at the time of recording. An example of a DNA melting curve analysis performed for food authenticity testing from the publication of Nixon et al. 2016 is illustrated here. The referred sample was a paprika spice, which was suspected of being contaminated with almond, an allergenic species of the genus Prunus. DNA was extracted from the sample and employed as a template for PCR amplification using a general Prunus real-time PCR assay. Additional species for the genus Prunus were also sourced and subject to DNA melting curve analysis using the same general Prunus real-time PCR assay. The resulting melt profiles were compared and, as can be seen, the profile for the referred sample contained a melt peak characteristic of DNA from almond being present, as well as an unidentified member of the genus Prunus.
As DNA melting curve analysis is a PCR-based technique, it is subject to many of the problems seen with real-time PCR. These typically include the late and sporadic amplification of samples, as well as significant reduction in the gradients of amplification curves. These observations are characteristic of PCR inhibition and can normally be rectified with dilution of the template. The presence of non-specific PCR products, including primer dimers, has also been reported for DNA melting analysis. This frequently occurs as a result of the use of non-optimal reaction conditions and can be rectified by optimizing the PCR reaction conditions. Additional problems commonly encountered include the generation of complex melt curves. These may result from the presence of unmapped mutations in the primer sites and require the redesign of the primer sets. Unclear or non-specific melt curves, characterized by curves consisting of zigzag lines, may occur when the presence of negative PCR controls have not been correctly designated during data analysis. In this final section of the e-seminar, a brief summary of the points covered is presented. In summary, DNA melting curve analysis is a post-PCR analytical procedure which employs fluorescent double-stranded DNA binding dyes in order to discriminate between similar DNA molecules which vary in DNA sequence composition. It may be applied to the analysis of samples of mixed composition by employing generic primer sets to a common DNA target sequence. However, the melting analysis procedure will detect all double-stranded DNA products generated during PCR amplification of a target, so careful optimization of the PCR reaction and preparation of DNA template is required. It is important to include suitable reference samples, if available, as well as appropriate PCR amplification controls. Continuing with the summary of DNA melting curve analysis, the method can be used with many existing and validated PCR primer designs and employed with many existing real-time PCR instruments. Because the technique is sensitive to the use of different reaction conditions and reagents, consistency in the setting up of reactions is essential. In addition, data quality is highly dependent on DNA template quality and target sequence complexity. Care should be exercised when preparing template and when selecting suitable DNA target regions for analysis. Finally, the technique represents a rapid, cost-effective approach that may be employed for use in food authenticity testing. We would like to acknowledge the support received in preparing, delivering and funding this e-seminar. In particular, the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2017 to 2020, the Department for Business, Energy and Industrial Strategy, the Food Standards Agency and Food Standards Scotland. And finally, we would like to thank you for your attention.